I am Muslat and moderator of this seminar. This seminar is organized by undergraduate students in Bioengineering department. I would like to start with a thank to our guest, Associated Professor Dr. İbrahim Tarık ÖZBOLAT, for not refusing us and accepting our invitation. It's an honor for us to be able to welcome you here, hocam. I want to share our outline. I will start with Associated Professor Dr. İbrahim Tarık ÖZBOLAT's biography, and then I will leave the stage for our guest for his presentation. It will take around 30 to 40 minutes. During the presentation, you can send your questions to my teammates via chat part. And when we are done with the presentation part, we will pass to the question and answer time. I will ask the questions to Dr. ÖZBOLAT, which you will going to send to my group mates. So let me start with the biography. İbrahim Tarık ÖZBOLAT is an Associated Professor of Engineering, Science and Mechanics, Biomedical Engineering and Neurosurgery, and a member of Haack Institute of the Life Science and at Penn State University. Dr. ÖZBOLAT's main area of research is in field of 3D bioprinting. He has been working on several aspects of bioprinting, such as bioprinting process, bioing materials, bioprinters, and post-bioprinting tissues, treatation of for manufacturing of the more than a dozen tissues and organs. Dr. ÖZBOLAT is a leading scientist with over 140 publications, including a sole authored book in his domain. Due to his notable contributions to the field of bioprinting, he has received several prestigious international and national awards, such as 2014 NSF Career Award, 2014 SME Outstanding Young Manufacturing Engineering Award, 2014 ASME Chow and Traeger Young Manufacturing Engineering Award, 2014 ASME Tau Pi Sigma Gold Medal, 2015 IIE Dr. Hamid K. Eldin Outstanding Early Career Industrial Engineering in Academia Award, 2015 International Outstanding Young Research and in Freeform and Additive Manufacturing Award, and 2017 Hazard Family Career Development Professorship at Penn State. And this is all. The stage is yours, hocam.
Okay, so thank you very much for the kind introduction, and thank you very much for the invitation. So it's a great pleasure to be here. So first, let me share my screen. So share screen, and you guys can be able to see my screen, right?
Yes, hocam. All
right. Okay, great. So as Muslat explained, so I have been in the field for a long time. As even the field's name was not really bioprinting at that time. So people were just calling different names like rapid prototyping, free-form fabrication of tissues, 3D printing of tissues, bioprinting of tissues. But later in the last 10 years, the field has been advanced significantly. So I think the bioprinting is now pretty much a standard name, but of course we have some efforts going on in the standardization of the terms as well. So what we do is we use 3D printing and then we apply that in the domain of tissue engineering. So we bioprint living tissues and organs spanning from bone to cartilage to skin to cardiac and pancreas. So we do that for various different purposes. So we manufacture tissues for regenerative medicine purposes. That means we want to have that implanted right, and then we expect that the implanted tissue restores the damaged tissue. We also have some tissue models like the lung tissue model. We have cardiac tissue model. We have pancreatic tissue or tumor tissue models that we can use those for different purposes, like drug screening purposes, or mainly for disease modeling. So how we can explore the disease models on these, mini printed tissues are really important. And there is huge interest on these as, using animal models. They don't really represent the physiology of humans. In the meantime, we can't really extract tissues from humans, right? So everything is pretty much dependent on two-dimensional cell cultures from human, but bioprinting has the ability to generate these complex organs like lung, right? Both the air-liquid interface, the barrier, so or the tumor, right? It has a lot of dynamic environment. So we use bioprinting to make this, but let's go through bioprinting first. But let's go through the agenda first. So today I'm gonna explain what 3D bioprinting means first, and then I'll move to the first section, which is going to be introducing a number of different 3D printing and bioprinting technologies and applications that we have demonstrated so far. And in the second section, I'll talk about the aspiration-assisted bioprinting, which is important for precise positioning of organoids and spheroids. And then in the third section, I'm going to talk about our recent publication on intraoperative bioprinting, so how we can use bioprinters in surgical rooms. So how can we directly bioprint the tissues on live subjects like animals? So bioprinting is a technique which is used for stacking and patterning of cells and other biologics, like it could be DNA, it could be microRNA, it could be proteins, in order to fabricate physiologically relevant tissues. So what you see is an animation of 3D bioprinting from a friend of mine, Christopher. So he basically developed this nice animation where it shows that inkjet printer, so it's the third dimensional version of a standard paper printer, is used to print the living cells, right, layer by layer with the support material, and a heart, right, a cardiac 3D heart tissue is being, or the organ is being built, right, with the blood vessels. So of course, this is just the animation, so it's just showing that the cells could be printed in this fashion, and then we can make an organ that's at the anatomical complexity of the native organs, as well as the functionality and physiology. So we're not there yet. So this is just the animation, of course. We're not at a point where we can make a tissue directly in the same shape, right, in the same function and physiology, and then we can transplant that into humans. So it will take some time, right? So in the last 10 years, we made significant progress. Nowadays, there are technologies that can basically anatomically reconstruct the shape, but it doesn't mean that it's 100% functional, it's 100% physiologically relevant. So it will take some time, but the field is making huge progress, and the recent technologies are very promising, and then hopefully in the future we're going to see the printed tissues in the clinics. So what we did is we did several different things pertaining 3D printing, bioprinting, for example. Here we did 3D printing of non-living materials like polycaprolactone, biodegradable materials, biocompatible materials. So these are like, for example, PCl, polycaprolactone, PLGA, and hydroxyapatite. We mix all these, and then we 3D printed those and make 3D scaffolds. So the goal was to 3D print these scaffolds for repair of cranial defects on the bone defects on rats. So we did the histology. We put these in rats, and then we kept them for 8 weeks, and then at the end of this period, we basically euthanize the animals, and we see that we got some bone regeneration with the composite material that we print with respect to just PCl, which is the clinically available polymer, right, and empty defect. As you can see here, we have bone regeneration that was better. We got some vascularization as well, which was promising. So the material is not really generating bone, but in the meantime, it was inducing some blood vessels. So, but we were not really super happy with the bone regeneration, right? What we see here is not really a fully regenerated bone. So then we work on some other aspects of the problem, and particularly the stem cells. So how we can basically have the stem cells better repairing the damaged bone. So in this regard, we use the red bone marrow stem cells, and we use the microRNA technology, the microRNA 148B, which induces or triggers the differentiation of the progenitor cells or stem cells into osteoblasts. So in this regard, we printed these polymeric scaffolds, and then we loaded them with collagen. Inside the collagen, we added the cells that are transfected with microRNA 148B, and we see that with the 148B transfected cells here in vitro, we got the cells, growing, they're proliferating, and in the meantime, they are differentiating osteogenically. So then we implant them into rats, and as you can see here with the microRNA transfected one in 8 weeks, we got pretty much like almost like a 100% coverage, which was very promising. So we also got a bridge formation. So that means we basically repair the defect all the way from the one end of the defect to the other end. So this was one study with the bone. Of course, now we do a lot of different work. We have a lot of different work with the bioprinting of bone, and most of the time we're trying to use hydrogels, not really polymeric, hard scaffolds. So in addition to that, we also work on PDMS printing. So this is a silicon-based material, and the printing of PDMS has been a problem. So in particular, this is used for organ-on-chip devices. So what we did is we basically use 2 different PDMS polymers. So the silicate 184 is the common one used in microfluidics, organ-on-chip device fabrication. So we mix that with AC 1700 in different ratios, and we made that 3D printable. We made different shapes. And then the good thing with this example is, particularly when we printed them, we see that without using any sort of proteins for cell adhesion, the cells were better adhering, with respect to just flat surfaces. So 3D printing was improving cell adhesion. In the meantime, 3D printing was improving the mechanical properties with respect to just cast samples. So we further work on that. So we made some microfluidic devices using this technique. For example, as you can see here, we made perfusible devices, and we line up with endothelial cells that makes the blood vessels. So we did that in 2D shapes. In the meantime, we created more complicated 3D patterns, using 3D printing, where we can, create 3D printing, 3D printed perfusible channels with the use of some sacrificial materials. As you can see here, we can basically control the patterning of this using 3D printing. In the meantime, the material is shear thinning, and it's something that we can easily dissolve, and we can perfuse that with different for different purposes. So we also did some other work with the embedded bioprinting, so which has been very popular these days. So embedded bioprinting is a technique which allows you to 3D print highly complicated, intricate shapes. Like as you can see here, we print the material that's in red with the cells into a support bed. So the support bed here is not really just supporting the 3D structure, but it also cross-links the printed material. So it's like a double materials, like one material is coming from the printed material and the other material is from the support material. When they interact, they cross-link and make a 3D structure. So using conventional bioprinting, like printing in air, like just on a flat surface, you can't really make highly intricate shapes. The beauty, the cool part of this technique is the 3D printed, the 3D printing of very complicated structures. For example, here we can make this kind of a very complicated cages, like a cage and a ball. As you can see here, there is a ball inside the cage, and the ball can freely rotate inside the cage. And these are not really something that you can make easily using, conventional bioprinting. So, of course, these are just some complex shape examples, but in the meantime, we printed some structures for medical applications like the bone, as well as we printed some cells like MSCs, the mesenchymal stem cells, and HUVECs, endothelial cells, the red ones. And we made some heterocellular structures that are very complicated in using this technique. So the other technique is a scaffold-free technique. So far, everything that I explained has been based on scaffolds. So scaffold is a temporary material that supports the cells, and then growth of these cells. In the meantime, the cells, they degrade these scaffolds. But we can also do things without using any exogenous biomaterial, any exogenous scaffold. So in this regard, we make a coaxial nozzle system. So the coaxial bioprinting is something that we discovered, and a lot of different groups have been using that in the last 10 years. So using coaxial bioprinting, we extrude some tubes that are semi-permeable. Then we inject the cells inside these tubes and we culture these tubes. And then later the cells, they aggregate, and then they got compact in size, and then their diameter reduces from 700 microns to, say, 500 microns. And then later we dissolve the capsule. So here's the tube. So once we dissolve it, the tube will be gone. So just the material, the cellular material behind it is gonna remain. So then we after dissolving that semi-permeable tube or capsule, we got these tissue strands in any length that we want. Of course, you need to have sufficient cell number. You can make it any length you want. Of course, the diameter has limitations, so it's about like 500 micron the max. Of course, going beyond that is going to impair your cell viability. So we printed T. So T is my middle initial, Tarık, T. And then this is the STM image. And in the meantime here, this is a nice photograph of the 3D scaffold-free tissue, which is like a mini sausage indeed. So if you make this using cells like smooth muscle cells, it can be a meat that's eatable, but of course it's extremely expensive. And then there are a lot of different companies now interested in 3D bioprinting or in general tissue engineering of artificial meat. Of course, the process is quite expensive at this stage. So we then further improved these tissue strands. So we impregnated some porogens. So we made it porous. As you can see here in the MRI image, the tissue strand can be porous. And here the goal with the porosity is to get better, oxygen inside, as well as the differentiation factors inside. So we use adipose-derived stem cells from clinics. So cells from liposuction surgery we got from patients. And then we differentiate them osteogenic and chondrogenic. So we try to make cartilage and bone. So we then put them in chondrogenic and osteogenic medium. We see that if we have the pores, then we better differentiate the tissue into osteogenic and chondrogenic ones with respect to solid tissue. So this was very promising. And then going back to solid examples, the solid strands. So we use the cells from bovine cartilage and the articular cartilage of bovine, so cows. And then we get the strands made, and as you can see here, this was positive to safran, no, other markers like collagen too. So here the important thing is when we look at the protein level, the SCAC content, which is the measured protein in cartilage, articular cartilage, was no different than the native one, so which was good. We look at the RT-PCR at the gene level, and then we see that the tissue strands were way better, expressing different chondrogenic markers with respect to just two-dimensional culture of cells. So then here we also demonstrated that once we put these two tissue strands together, they fuse. So we made them from using different fluorescent markers, and we'll let them fuse. And as you can see over time, the cells from different tissue strands, they cross-migrate the boundary, and then they fuse and make a single patch of tissue. So we use this principle, and we try to apply that for bioprinting. So in this regard, we use some interesting bioprinter hats using a 3D printer. It was from Envision Tech. So we made high resolution, high definition nozzle structures, and then we put the strands inside, and then we extrude them. And then we also made some interesting cartridges, which is something that you can replace during the and for example, when you need more tissue strands, you can plug in neve or load neve cartridge during the process. So using these, we printed some cartilage tissue strands next to each other, and then in 3 weeks they make a patch, and then when you look at the patch, histologically very close to the native one. So this was very promising, and it basically, you know, enabled us to make an articular cartilage patch, and which can be used for, you know, cartilage repair. So recently, we further advanced this technique, and then we tried to make articular cartilage more, you know, physiologically relevant articular cartilage. So the major problem with the previous system was we could make only a single layer mainly, and when we look at the articular cartilage, you know, proteins like collagen, this is one of the major proteins, is heterogeneously distributed in cartilage. So the top layer of the cartilage is the collagen is more horizontal, and for the deep layer of the articular cartilage, the deep zone, it's perpendicular. So the goal was here is, you know, how we can recapitulate that organization of collagen. It's a beautiful organization, which has been, you know, highly challenging to recapitulate. So in this regard, we look at these cartilage strands, and we notice that the collagen was longitudinally oriented with respect to the cartilage, so strand. So for example, if this is a strand, the collagen was oriented longitudinally in this way. So using this property, we basically printed the tissue strands vertically, which has been a new technique, right, using aspirationless bioprinting. So it's a new technique that I'm going to explain in the next slides. And then we printed them into an area of pin, okay, not really skewering them, but it's in the space between the pin array, so that they fuse. And then on the top, we printed the layers, you know, the superficial layer horizontally, and we combine them. At the end, we got the tissue strands, you know, all assembled into a single patch of cartilage, where the, you know, collagen orientation was more perpendicular in the bottom zone, the deep zone, and then more horizontal in the superficial zone. So we use these tissue strands and then made also perfusible, you know, vascularized tissues. So in this regard, we use a multi-ion bioprinter that I myself and a former PhD student of mine, we developed together. So this is a multi-ion bioprinter which has the strand printing apparatus as well as the coaxial nozzle. So the coaxial nozzle is a technique. Coaxial bioprinting is a technique. Of course, coaxial nozzle is just an apparatus. So coaxial bioprinting is a technique which enables the deposition of tubular structures, you know, directly, straight from a nozzle tip. So we discovered that, you know, back in early 2011-12. So at the time, we showed that using this technique, we can extrude the alginate, and we can also extrude the calcium chloride in the core, and then we can make a tube that is 3D printable. With the use of Bioprinter, we could be able to control the deposition pattern of these tubes. Of course, we can have the cells laden in the alginate tube. So using this technique, we made some perfusible blood vessel constructs, and as you can see, this was the middle one was longer than a meter. So this is a branch one. So the branch one here is was done manually. So it was very small with respect to a penny. It was very small, so we have the cells growing inside, no problem. In the meantime, we integrate that into larger, you know, hydrogel constructs. So we perfuse that, and then we kept the cells viable inside. So this has been, you know, featured in Popular Science back in 2013 as well. So using this technique, we combine this with the strand printing. So we print the vasculation strands, and over, you know, a couple days, we expect that they fuse and then make a tissue that's circulating the blood vessel. Of course, this was a cartoon in our vision, and then we tried that experimentally. So we printed that around the vasculature. So this is just a temporary mold that we kept it for a couple days until the tissue strands self-assemble. So then they self-assemble, and as you can see, when we cut it, we see that the tissue strands, they fully fuse, and then they enclose this vasculature. So we use, of course, different perfusion systems and make vascularized tissues for for creation of tissue models in a little chip. So one example was to use a perfusible chip that we can basically load the different, tumors or pancreatic islets or, different organoids, and we can make them perfusible. And we expect that after fabrication, we can line up with endothelial cells, and these, organoids or spheroids, it could be like tumor spheroids, they can sprout out capillaries and eventually they'll connect, and we can perfuse some immune cells that are engineered. So I work with, different people, different experts from different universities, like immunologists, like Dr. Unutmaz from Jackson Lab. So we then use these 3D models. So we show that once we print or once we load these tumors in the 3D constructs of gels, we can easily see that the cancer cells, they grow. The green ones are cancer cells, the red ones are endothelial cells making vasculatures. As you can see, they sprout out capillaries, and then we eventually see that some of the cancer cells, they actually invade into these tumors, and this can be used for metastasis studies. So we have now some chip studies going on, so we can have these tumors that are loaded with control distance from the perfusible channel, and we can perfuse this channel, and then we can have these cancer, the tumor spheroids, they sprout out capillaries which are anastomosing to the main perfusible channel. So this is still ongoing. And recently, to make the tumors or any organoid to be printed precisely in gel systems, we invented a technique called aspiration-assisted bioprinting into gel systems. So here we can use that in multiple ways, either a scaffold-free way where we can print a dissolvable gel like alginate, and then we can cross-link that, and then after cross-linking this, we can print the spheroids. So what we do is we pick the spheroid using a pipette tip, and this is automatically happening. So then we go and load that on top of a support gel, and then we can do that precisely, right, by layering different spheroids, and then we can make this kind of a 3D pyramid. And then over time, in order to assemble them, we need to have another layer of a sacrificial gel like alginate. So then after some time, like when the spheroids is self-assembled, then we decross-link the gel, and then it leaves behind the self-assembled tissue of spheroids. So this is one approach. Of course, there are other approaches where we can use this system in the functional gel systems. For example, here we're printing fibrin. So fibrin is a functional material. Functional means it's a scaffold. So then we cross-link fibrinogen and thrombin layer by layer. So the blue one is thrombin. So we make 3D fibrin structures, and then we go and then print the spheroids, and then we can print them precisely, different types of spheroids at different locations, and then we can then get them crosstalk. So they can sprout capillaries. We can make blood vessels out of those, and then in this paper we actually look at how the blood vessels from different spheroids, they crosstalk. So of course we did some engineering analysis on this, and we try to understand how the spheroids needs to be picked. So we look at the physics of the problem, and the critical thing here was, I'm not going to go into details of all these equations, but the critical thing was we need to have a sufficient pressure in order to lift the spheroids from the media, because there's a huge forces here acting because of the surface tension between the cell media, tissue, and air. Okay, so basically the surface tension is pulling the spheroid down, so we need to have the spheroid going up. Of course, this was important. If this pressure is a lot, then you can aspirate the entire spheroid. If it's too low, then you can't really be able to lift the spheroid. So in this regard, we tested various different spheroids, from HUVEC to fibroblasts to cancer cells and human combination of different cells like MSCs and HUVECs and MSCs. As you can see here, is the surface tension, which is a mechanical property, increases as you go from left to right. The surface tension increases. In the meantime, the compactness increases, right? In the meantime, the roughness decreases. So as you see here, MSCs are less rough versus the HUECs are extremely rough. So we determine the critical pressure to lift this, and it's because when we apply too much pressure, then these tissues are actually viscoelastic. So that means they're both viscous and elastic at different, time points. So they can be viscous or elastic at different time points depending on the stress that you applied on them. So at the beginning, they act like more elastic. But if you keep the same pressure and apply that long time, so then you basically change the behavior of these spheroids from elastic to a viscous regime. So this time was here is the relaxation time, which is a critical time. So you should basically print these spheroids before the relaxation time, so that you keep them in their solid state. So we basically determine the relaxation time for different materials, and relaxation time we print that in most like 20 seconds, 15 seconds, and at that time frame was very sufficient to print these spheroids without transitioning them from solid to liquid phase. So using this technique, we printed spheroids in various different shapes. Like, for example, we can make them a line, we can make them a triangle, we can make them in, some heterogeneous structures like this using different spheroids. They were labeled with different colors here. The blue is the big one, red is the smallest one, right? And there's also the green one. So here the blue, the red here, you see that it's not really spherical, so it's some irregular shape. So this technique also allows us to print some irregular shapes. So we print these spheroids onto different gels like gelma, gelatin metacolate, or collagen. As you can see here, we made a ring. So we eventually print them on top of each other. For example, we made this structure using 8 spheroids and printing on top in the air without tipping them over, so they don't really collapse. Right? We call that as like a pizza tower. So the max that we climbed was just the 8th layer. So at the 9th layer, unfortunately, fell down, but 8 was very successful. So we did bridge configurations, different examples using this technique, and then. As I explained in the previous slide in the animation, right? So we basically printed spheroids close to each other, and we see that how these spheroids sprout capillaries, make blood vessels. And as you can see here, we can control the distance precisely from 400, 800 to 3000. This is a control group. They were extremely separate. They were far away from each other so that they don't really have any interactions. But when we look at the 400 and 800 micron, we see that there's a lot of interactions going on. As you can see here, the sprouts from different, spheroids, they anastomose, right? The blood vessels, they join. As you can see here, there are some blood vessels made of 2 different cells. So then we look at if there is some sort of a directionality. That means, is there any impact of having a spheroid next to the spheroid of interest? So, for example, here we did the directionality analysis when we put 2 spheroids. If there is a neighbor spheroid, right, and we see that the sprouts, the capillary growths from one spheroid are mostly towards the other spheroid. Okay, we see that there is directionality. And this was an interesting example. And we did some other interesting examples like, spheroids made of different cell types and different sizes could be also printed. As you can see here, like bigger spheroids in the middle versus the smaller ones in the 1st and 3rd layers. And then we use this technique to make, in one of the recent publications, we made that for bone regeneration. So in this purpose, we use MSC and HUEC spheroids. They were combined together, and we osteogenically differentiate them to make bone spheroids, and we printed bone spheroids. And we did the micro CT, and we see that at each layer we got some bone deposition, over time. So, and then in another publication, we show that using this technique we can make osteochondral tissue, so made of both bone and cartilage. So this could be used for drug testing purposes or for disease modeling, even like for regenerative medicine, if we can make some larger patches. So in this regard, we start with ADSs from clinics, adipose-derived stem cells, and differentiate them to osteogenic ones. As you can see here, this is the osteogenic one with huge, bone deposition and calcium deposition. So this is the other zone red staining. And then we also have some, cartilage spheroids. As you can see here, these are the cartilage spheroids, right? So they were positive to ASCAG. So then we printed them precisely. So 3x3 arrangement in the first layer and 3x3 in the second layer. And then we print the osteogenic and chondrogenic ones in the first and second layer. And then we made these, interfaces where we have the bone here on the top. The dark one is the bone, and the bottom one is the cartilage layer. So this was an interesting application. Actually, we're working on another manuscript now that we are trying to do the differentiation without using differentiation media, but rather than differentiation media, we're now using the microRNA technology, like the I think the mRNA technology is a huge interest, right, because of the COVID-19 vaccines, right? Here, right, we have two major vaccines from mRNA, but so we use a microRNA. It's not mRNA, but microRNA. And then we created. We're now creating this implantable osteochondral tissues. So we then use this technology in another publication, recent publication, that was highlighted in the Genetic Engineering magazine. So in a previous technique, so we were printing into gel, right, onto gels too, but the major problem is we can't really, make the manipulation inside the gel easily, because there has been some limitations where the gel needs to cross-link, and it needs to have some yield stress properties or, self-healing properties. So then we attempted to use self-healing hydrogels that are yield stress gels. So the yield stress gel means once you apply, and I just want to show this in the video. So here, this is the liquid compartment. So we basically pick up the spheroid from the liquid compartment. Here, this is the media compartment. And then we take it and then move to a transition phase here from the media compartment to a gel compartment. So here the support gel is yield stress gel. So that means when you take it and move, as you can see here, it moves easily. You can position that wherever you want, right? You just leave it and then put it, and then it will stay there. So here the beauty of this technique is like you leave this spheroid and it stays there. So here the yield stress gel is a gel. When you apply a shear stress, then it basically acts like a liquid at this region. And once you stop the needle move, then it becomes like a solid gel. So it basically keeps the spheroid at the position. So we did this technique, and then we pattern the spheroids in the way that we want. In these support gels, could be carbopol or could be alginate microgels that we prepare in the lab. And then we created 3D structures. For example, we made this helix shape, which hasn't been possible in the past, right? So we made these cylindrical shapes, and we expect that these spheroids they self-assemble and make a tube. And then we made more complicated structures like a DNA strand shape using two different spheroids, like small and large ones. Okay, so far I demonstrated more like in vitro examples, right? Some with in vivo, but mostly in vitro, where we created tissues, and then we just use them for in vitro analysis purposes. We also have some efforts going on in the translation of the bioprinter technologies into clinic. So this is the goal. The ultimate goal is, and one day we would like to see the bioprinters in clinics that are repairing the human tissues and organs. So in this regard, we use the intraoperative bioprinting. That was also known as like in situ or in vivo bioprinting, where the tissue can be bioprinted directly into a defect in a surgery. So we further advanced the existing work substantially, and then we basically created composite tissue. So what we did is we created defects on rats, 2 defects, and then these defects were all the way down from the epidermis to dura, so close to the brain. So we basically try to regenerate that using 2 different materials called HT ink, the hard tissue ink, and ST ink, the soft tissue ink, and we print various different cells, proteins, growth factors, and differentiation factors, and cross linkers. and then we use 3D scanner to scan the image, and then we first print the bone using the HT ink, the hard tissue ink, then a barrier layer, and the dermis and epidermis. So this was the skin, and this was the bone, and the barrier was like an interface. The reason why we put the barrier is because we really want the cells in the dermal region not really migrating down because of gravity. There is a sedimentation of sedimentation. That means like the cells, they basically go down, right, because of gravity. And then if they do that, then they can go to bone region, and then they can generate soft tissue in the bone that we don't want. So that's why we put a barrier. But in the native tissue, this is called a periosteum, a very tiny tissue. And now in our current efforts, we're also trying to replace this with the physiologically relevant tissue. That's the periosteum, which is rich in stem cells as well as blood vessels. So we created the hard tissue ink. So hard tissue ink is made of collagen, chitosan, hydroxyapatite, some other ingredients, but mostly collagen. And then we printed that, and this is an extrudable ink. And as you can see here, we made grease structures, and we print the cells from the rat bone marrow stem cells. And then as you can see, in day 7, the cells, they grow crazy. So that means the cells, they really like this material, and the material is osteogenic. Indeed, we did the differentiation study. So we printed these structures, and then we osteogenically differentiate them. And then the RunX2 and BSP, these are osteogenic markers showing bone deposition early and middle and late stage markers. So as you can see here, we also did RT-PCR, but not really going in full details. But what I want to say is, without using any media, okay, without using any media, the HT ink just supports osteogenic differentiation of stem cells. So that means we don't have to worry about this after printing. And we printed the HT ink as well as HT ink plus recombinant BMP2. Recombinant BMP2 is a clinically available material used in bone regeneration. And empty defect, as you can see here, at 6 weeks, 5 millimeter defects on rats. We got about 80% bone coverage area for the last group. So that was very promising. So we didn't really use any cells. We didn't really use any sort of like, genetic modification. It's just the material itself was making that. So then we also work on the skin printing. So for skin printing, we work on the droplet-based bioprinting technique, because for extrusion, it was not really ideal for the skin regeneration, as the skin has very thin compartments. So that's why we use droplet, which has better precision, and so the resolution of the system was better. So we print droplets, and then we look at different materials. So the ST ink, the soft tissue ink, was made of fibrinogen and thrombin, and we crossing that with fibrinogen and collagen, and we crossing that with thrombin. And we did a lot of droplet analysis, and we see that the optimal, we try to find the optimum, parameters for generating droplets that are small enough. So the droplets were about a couple hundred microns in thickness. And then we printed these, and then, we see that in particular rat dermal fibroblasts that we print from same rats, we see that the cells, they grow and they really like the material. And then we therefore, use this material, prefer to use this material for skin printing. So for skin printing, we intraoperated printed that using the droplet-based bioprinting system. So we created defects. So this time the defect size was 6 millimeters. So bone defect was 5 and skin defect was 6 millimeters. The reason we want a larger skin defect is because we could be able to make that the surgery easy. In the meantime, we'd like to do scanning as well. So then we 3D printed the skin tissue. So we had 5 groups. So the native skin group, we have the negative control, that's just empty. We have the ST ink, soft tissue ink only, no cells. And then we put cells for the other group. And then this was the most complicated group. So ST ink with the red dermal fibroblasts and KGF on top. So we basically print this one for the dermis, and on top we put the keratinocyte growth factor for the epidermis to generate the epidermis and re-epithelize the epidermal region faster. So we see that the printed groups, and particularly the ones with the KGF, it has the fastest healing, reepithelialization and wound healing. And then we look at the mechanically as well. From the mechanical point of view, we have this group generated the highest mechanical properties, but still less than the native skin group. And the total timeline was 1 month, 4 weeks. And then we also did histological analysis, and we see that the printed groups, they basically turn into all the groups. Actually, they regenerate the skin, because the skin regeneration is not really a super critical, a challenging task, but the bone regeneration was a challenging task. When you add the skin on top, then it becomes extremely challenging. So here, skin-only defects, they healed, but the printed ones, they healed better, and more collagen deposition was observed with clear distinction of the epidermis and dermis layers. So then we combine these two techniques, bone printing and skin printing, and make the composite tissue bioprinting in reality. So make it a real. So the surgical approach is mainly, using the bone from some bone bank, right? The external bones, like allogenic bone, or it could be, the bone from the same patient, if there is, sufficient bone in the patient, that is harvestable. And then on top of that, there is a muscle tissue. So the surgeon doesn't really put the skin; they just put a muscle to make the bone vascularized, to keep it vascularized. So eventually, they expect that skin grows on top of the muscle. So here, our technique will enable the reconstruction at once. So we start with the bone printing, and on top of that, the barrier layer, and on top of that, the dermis, and on top of that, the epidermis, right, layer by layer. In 4 weeks, we got the skin regeneration. It's pretty much, full closure. And in 6 weeks, we got better bone regeneration with respect to week 3. But the bone regeneration here was about 50% coverage area, which was less than the bone-only defect. Of course, the composite defect, the bone compartment for the composite defect was directly open to atmosphere. So it was extremely challenging task. And there was like less vascularization. We identified vascularization as well, but now the next step is to further advance this, adding more, physiologically relevant layers as well, like periosteum, having the hypodermis part of the skin in the meantime, enhancing the vascularization in these tissues. So the next step for the intraoperative bioprinting work is something that we would like to see on the large animals. So in this regard, we actually started working on sheep. So this is just showing a pig model from the advanced solutions, but we are trying to regenerate the bone defect on sheep using a 6-axis bioprinting mechanism, because the previous ones are just fine for small animals like rats, but for large animals, we can't really use just like gantry type of 3D printers, like 3-axis. So in this regard, we would like to use like robotic arms. Of course, we would like to have these technologies translating into clinics, which was the cover image of my book. So this was the vision. So how bioprinters, can repair the human tissues. It could be for plastic surgery, could be craniofacial surgery, for various different things. Can be done using this technology. So with that, I would like to thank everybody for their patience and for their attendance, and I would like to acknowledge the people that has contributed to this work. Almost like 50 lab members contributed to this work. In the meantime, I have many collaborators, so they have been extremely helpful for providing various different expertise and technologies, or even like biological materials. So we are here at Penn State, which is the most beautiful building on campus. So we're on the 3rd floor, and our research has been funded by various different agencies. So with that, I'd like to conclude here, and I can get your questions, if any.
Okay, can we share the screen, please, Ali?
So I'll stop my
Yes.
share. Okay, there we go.
Okay, first question. In most of 3D printers, one of the main problems is about supporting structures in printed body. They require post-process work, and while removing them, it may damage the body. So in 3D print bioprinting, do we have such a problem? Do you have any alternative solutions to remove these supportive structures? Also, there are some 3D printers that are working more than 3 axes. Is there any possible to use 5 axis or 6 axis 3D printers in bioprinting? Thank you.
So I think I already answered the last part of the question. So there are, of course, you know, higher degrees of freedom options, like a 6-axis bioprinter that we already started that work on sheep. And in the lab, in the past, we also developed 5-axis bioprinter. I mean, the real surgery, the surgical robots are, of course, they're higher degrees, right, like 6-axis. So our goal is not really just use a 3-axis stuff for, you know, for larger animals, and eventually for human use in the future, that's gonna be like more advanced, you know, printers. Of course, what we did in the body, I mean, it was you know, without any support, so we didn't really use support material because we were printing inside the defect. So the defect itself, the body itself, was mainly supporting the structure. But if you want to, you know, create something outside the body, right, that's kind of porous, or that could be, you know, in the shape of the organ that you want, and then transplant that, yes, you need to have a support layer. And I think we already, I already show an example where we created something in the support material that enables, you know, creation of very complicated shapes. But at this point, I can't really use that for intraoperative printing. In the future, it could be, but at this point, we haven't really demonstrated that.
In what position do you see in the future of bioengineering, which is shown as one of my future professions?
So the bioengineering is a very broad term. I mean, you know, each aspect of bioengineering could be medical device technology, could be biomaterials, you know, 3D bioprinting, or other disciplines under the term bioengineering. But for the tissue engineering, regenerative medicine, and bioprinting, I see, like, you know, fabrication of more complicated, like, solid organs, physiologically relevant solid organs, and use them for transplantation in humans is going to be the future. And, you know, I can easily compare that with respect to what has been accomplished in the last 10 years versus like today. You know, 10 years ago, we could be able to just print cells mostly, you know, make less complicated shapes, you know, but nowadays we can really make highly complicated, intricate shapes. In the meantime, we advance the technologies. In the meantime, we kind of had a lot of understanding about the biology behind the tissues too. So now we have some, you know, volumetric structures that have been printed and implanted in animals. Hopefully in the next 10 years, we'll see more, you know, translational efforts. So if you're interested in particularly Sena Sude, I mean, she can be interested in the clinical translation of all these efforts. I think there could be a lot of potential.
How can we keep those receptor cells alive in a study on pain, pressure, and hot and cold sensing receptors in skin tissue production in collagen matrix? Also, how can we measure the sensitivity values of these receptors in artificial skin tissues? Thanks for the answer and presentation.
Well, we haven't really focused on the receptor cells so far. So we had some interest in the use of in creation of three-dimensional intestine tissue for opioid use. So, you know, how can the receptor cells, you know, better sense the, you know, opioid and particularly we were also interested in the blood-brain barrier with these receptor cells. But for the skin, what we have done so far is we try to make all the conditions, you know, at the physiological temperature. So we didn't really make it hot, we didn't really make it cold, or we didn't really apply any sort of pressure. So we basically didn't really have that factor in place. But the sensitivity values, to measure the sensitivity values of these receptors, I think one important fact is there is a huge progress in the variable, you know, sensors, right? We're also interested at Penn State. We're now hiring a faculty member in the area of biosensors. So we have seen many work that's just, you know, using these variable electronics that can sense, you know, different things on the skin. I think I mean, those are mainly on the skin, but if you can use some, say, skin patches, like a little needles that can be inserted in the skin, and then somehow, you know, try to kind of sense the receptors and their function, test them, you know, measure them, could be one way to make those measurements. Again, I'm not really expert in variable electronics and sensor, but you know, there is some work in this area that has some potential.
Ali. What kind of support materials are used in your studies?
So the support material, we use carbopol. For spheroid printing, we use Alginate Microparticles. So carbopol is a microgel. It's commercially available. Alginate Microparticles is something that we made in a lab. So it's like little microparticles of alginate at 50 micron. They're suspended in just media, or it could be PBBS. You know, there might, and for the 3D printing of the suspension printing of using extrusion technique, we actually use alginate microparticles for the support pad. So alginate is something that we can easily decross-link, and we can easily liquidify. But there are other support materials people use, like gelatin microparticles, you know, other microparticles. So there are some commercial gels, some gels that could be synthesized using like gas host type of gels that have been demonstrated in the literature so far by different groups.
Okay, 2 questions. Do you have any studies with 4D bioprinting? What do you think about 4D bioprinting studies and its future? And the second one, what are the techniques and materials used to reduce cell damage when printing with cells?
So I'm not really involved in any 4D bioprinting efforts. So this is a very interesting concept. I actually proposed that concept back in 2014 in an opinion article. And then sometimes people think that in opposite way, that you can propose, like they think that this is not really, it was extremely speculative. So, but later several different groups show different examples. So we don't really have anything that, you know, where the shape change was critical, but I can see that the major challenges, you still deal with soft tissue gels and for bioprinting, and then it's really tough to control the shape as opposed to some polymers, like hard polymers used for 4D printing. So more gel needs to be synthesized, and I expect that more tougher gels, like more elastic gels, could be synthesized for bioprinting purposes. So the other problem is, of course, once you put that implant, right, there is also the biodegradation. So it's a very complicated problem, you know, between the, you know, the gel domain, right, the degradation domain, in the meantime, there's huge mechanics involved. So regarding techniques and materials used to reduce the cell damage, we don't really have any sort of problem with, printing cells that are not really going alive, but there are techniques, of course, where the cell viability could be at different levels, like extrusion, droplet, inkjet, microvalve, laser-based technique, or stereolithography-based techniques. These are all different depending on the techniques. Of course, the materials is also important in the fact that super viscous materials can't be really printable, and sometimes they may harm the cells. In the meantime, these materials could be toxic, and it could basically have a detrimental effect on cell viability after printing, not just during the printing process. So if most of the time we don't really see issues with the cell viability, but if the cell viability is a concern, then we need to optimize the printing parameters, say try to increase the nozzle size, reduce the printing pressure. So these are some of the ways to improve the cell viability.
We have a lot of work in making vascularized tissue. What are the key challenges preventing clinical translation? Where is the bottleneck, and how can this be overcome?
So the vascularized tissues, of course, there are a lot of different challenges towards clinical translation. So one challenge is, the level of vascularization. If the vascularization is really stable, without any occlusion, without any sort of, problems. Sometimes the vascularized tissue may not really, the vascular pattern can't really anastomose efficiently to the body. Sometimes there are some occlusions, right? Thrombosis is one of the other factors, so you need to basically line them up with endothelial cells. Otherwise, you can't really get, you can't really make it functional over time, as it can thrombose easily. Other than that, the other major challenges is not really just vascularized tissues, I can say, but any tissue so far, right? It has huge. Although they have huge potential, one of the critical aspect is, you need to go through the FDA pathway, right? Anything that involves cells is really hard to translate into clinics because of the involvement of cells, right? There are a lot of concerns, raised by FDA, right? The source of cells, right? The viability of the cells. The purity of the cells, the media or the culture environment that you grow the cells, how you're gonna, and the environment that you'll do that, like, is it gonna be, outside the clinic, or is it gonna be just next door in the clinic where you're gonna isolate the cells from the humans? So I think it's not just vascularized tissue is a critical problem, but overall, everything that involves cells has some regulatory issues at this point. But for vascularized tissues, again, the critical thing is your vascularized tissue functional over time? If you show that on animals for long term, that's going to be encouraging, but you can make a vascularized tissue in vitro, but it may not really stay functional after the implant.
In 3D printing, what are the strategies to make scaffold mimicking the host tissue's behavior even in the body environment in vivo?
So I think this is a good question. Very relevant work has been done, in particular in the last 5 years. So one of the important factors, we use different gels, and each gel has different properties, and several different companies, they're trying to make universal materials that can be used for various different organ and tissue regeneration purposes. But the problem is there's no universal gel that's gonna promote tissue regeneration for every tissue. So every tissue is different, every tissue environment is different. And in this regard, the decellurized ECM based bioinks have been extremely popular. So what you do is, say, if the tissue of interest is skin regeneration, so then you can get the skin harvested and decellularize it, remove the cells, and then chop that into pieces, and then use some detergents like some enzymes and basically get the ECM out of that tissue. So using this ECM, so that ECM basically carries the natural cues, right? The native cues, and then you can use that for skin regeneration. There have been several tissues done using this, like liver, skin is one type, even like cancer, blood vessels, and with the use of these, we can better mimic the host tissue behavior.
Okay, 2 questions. Can we use bioprinting extensively for artificial meat products? How can we make it affordable in the public? Are there any potential hazards resulting in its long-term consumption? Regarding bioprinting for organ transplant, the specialized cell that we make from the patient are from his or her embryonic cells, isn't it? Could it be possible to take it from other sources and yet make it the body accept and tolerate the foreign object? Thank you very much for your time and consideration.
So let's start from the second question. I mean, the use of embryonic stem cells has been demonstrated, even like we use embryonic stem cells in the lab too, but embryonic stem cells has other issues like regulatory issues as well. So we therefore stick with other stem cell types. I think the most convenient one is the induced pluripotent stem cells derived from skin. Basically, you get a skin fibroblast, which is very easy, just a small biopsy from the skin. You get the fibroblasts, reprogram them into stem cells, and use them for different tissues and organs, which is widely used nowadays. So of course, regarding the rejection, you can't really use someone else's stem cells to other people easily. So one of the important direction is now using the universal cells or creating universal stem cells, so that you can basically when you implant that to some other person, that person can easily tolerate that foreign body reaction. So it can accept that as his or her own cells. So this universal stem cells is now is one of the important areas that I have some friends in stem cells, doing that kind of work. And there is for the bioprinting the meat, I know there are multiple companies, and the major concern was to make it affordable. And so far, the tissue production that we do in the lab is quite expensive, right? We need to culture those with some expensive media. So in this expensive media, I mean, this is one fact. The other fact is, culturing the sufficient number of cells. So if you can make everything in a more affordable way using, more or like a mass production using large bioreactors without using, serum in the media, make it more affordable, you can decrease the cost. But I'm not really involved in this, so I'm not really involved in any sort of research or any collaboration with companies. And I don't really know, the cost level of these things. So of course there are multiple companies. They are claiming that they had some, efforts in making everything more affordable, but it's not my expertise. But regarding the potential hazards resulting in the long-term consumption, of course this is very speculative at this stage. But anything that you use during the culture of these tissues might have some side effect in the future, and again, it's very speculative at this point.
Okay, what is the benefit of using separates instead of scaffolds for bone and cartilage regeneration?
Yeah, so this is also a good question. With the scaffold, the main problem is degradation, and you can't really print the cells at cell densities close to the native tissues. So, I mean, with the bioprinting, we in general print cells in gels, you know, ranging from say 1 million to like 10 million. Sometimes we would go 20, 40 million. But if you want to do say 100 million, 200 million cells per ml at the physiological, you know, density, cell density, then spheroids could be better. So, and then of course, there is a degradation problem with the material that you use. Sometimes the degradation products could be toxic to cells. Sometimes cells in the scaffold, they can't really communicate well because they're too, you know, far away from each other, and they can't really interact. But when you put the cells close to each other using in spheroids, right? So then the cells, they better communicate. So this might result in better, you know, outcomes, and particularly for cartilage regeneration. You know, spheroids has been very promising, and then there are some examples. For example, there's the spheroids of cartilage that have been tried in human you know, research, like human trials in Europe. So using cartilage spheroids for also control defects. For bone, of course, there are some limitations of spheroids, but you know, so far we have made quite a bit of progress, in particular with the use of endothelial cells in the bone. So the major advantage is, of course, the cell density, you know, improved cell-cell interactions, as well as you know, mitigated issues because of you know, no degradation-related problems.
Okay, Hocam, I will ask my question right now. I have two questions. First one, how and where does those externals are manufactured? Can we manufacture a similar bioprinter by ourselves?
Can you repeat the question, please?
Yes. How and where does those extrusions are manufactured? Can we manufacture
storage?
Extrusion.
Extrusion, huh?
Yeah. Manufacture a similar bioprinter by ourselves.
You mean, can you 3D print the extrusion units?
Yes.
By yourself?
Yeah.
Well, it depends on your application. So if you just use standard application, I think there are already nozzles. So you don't have to worry about, you know, making those nozzles. But if you have something very specific, very complicated, sophisticated, that is not really commercially available. Yes, you can use the 3D printers to make these nozzles, but the 3D printers may not really give you something very precise. So that's the problem. You can't really make a high resolution structures using 3D printers. If you want to get, say, a resolution of, say, 100 micron, 150 micron, so I suggest you don't really make anything 3D printed. Just go with the commercial options.
Okay. For the last question, and it's mine, what are the essential skills to have a career in 3D bioprinting?
So the essential skills in bioprinting, number one, you need to have biology background. So you need to be able to culture cells and grow cells, and you know, culture, at least have expertise with culturing, you know, mammalian cells. And number two is have some experience with biomaterials. So you need to have some hands-on experience with using different gel systems, you know, how gel system works and how you enclose or you know, load the cells in the gels. Number three, you know, background in some mechanical engineering with the use of 3D printers, you know, hands-on experience in just, you know, manipulating some bioprinter parts, like making those parts, or at least, you know, assembling those. So if you have these three expertises, somehow, you know, you'll be very successful. Of course, there are things that you can definitely have some more expertise with the animal work, you know, your expertise in handling animals, doing surgeries, not, I mean, leading a surgery, but at least, you know, involved in surgeries could be helpful for us.
Okay, I think we have to finish the question part because we're kind of running out of time.
That's okay.
There were many more questions, but we can't take it anymore. This is the end. Before we ending this seminar, we would like to share something, Hocam. Thank you for estimating. We thank our estimated guest, Dr. ÖZBOLAT, for his presentation. And we have donated on your behalf to MiHave platform. This is a platform founded by Sena Sude Taşkın, who is in our crew. The platform's aim is help treat and feed the animals who's needed, and we donate it for yourself.
Okay, thank you. Well, I appreciate that for this, you know, nice certificate. So it was a pleasure to, you know, share my share our recent findings, and most of the things that we've done is in the last couple of years. So I believe, you know, everybody, all the audience, could at least learn something about bioprinting and the recent state of the art. So it has been a pleasure with you know, staying with you guys for a couple hours and sharing all our recent findings. And I hope you guys will continue, you know, hosting other speakers in the field of bioprinting. So this is very important for advancing the field of bioprinting.
Okay, thank you.